Welcome, everybody. This is the April 3rd meeting of the working group and GLIMPRINT. We have a single talk today. As always, these meetings are live streamed and recorded on YouTube, so be aware of that. Tom, myself, Jim Sluka, and Lorenzo are always available to take your suggestions, comments, ideas. Please feel free to reach out to any of us with suggestions or critiques. We have a variety of communication channels. Things work better if we use them. There's been some discussion about discontinuing the X/Twitter channel, but we have the other channels available, and we'd appreciate your help distributing announcements for the group. If you have a publication to share, we can share it both on the IMAG working group page and also on the GLIMPRINT website. Please, anything that you would like to see distributed that you've written that's relevant, send to jsluka@iu.edu, and we'll make sure that that gets publicized for you. The GLIMPRINT page allows you to post seminar announcements, meeting announcements, job announcements, and so on. Please take advantage of that. Again, if you have any issues with that, jsluka@iu.edu can help you out. Tom Helikar as well can do that. So please help us populate that site. The plan at the moment is to make life easier on our European colleagues and less easy, I'm afraid, on our West Coast colleagues, to move these meetings to 10 a.m. Eastern Time, starting on the 15th of May. And that will allow the Immune Digital Twin group through our DA, led by Anna Niarakis, to participate more fully. If it doesn't work, we can move the times back, but currently, effective May 15th, the plan is to move these meetings earlier. A few announcements. We have the CompuCell virtual tissue modeling workshop coming up in July and August of this year. If you want to sign up for that, you can go to CompuCell3D.org/workshop25, and you'll find the information there. It's going to be a 2-week workshop this year. We definitely hope people will participate and let people know about that. This year we are charging a $25 registration fee, although that's waived in cases of hardship. But if you're interested, contact me, talk about that. There's going to be a cytoskeletal modeling workshop in Raleigh, North Carolina, May 12th through 16th, led by Giulio Belmonte at North Carolina State University. I believe it says registration deadline is March 30th, but I believe they're still taking registrants. If you're interested, that's a slightly different scale of modeling from the one that we normally work at in this group, but it's a very interesting modeling package that could be of interest to people in this community. We have the 2025 Open VT Reproducible Agent-Based Multicellular Modeling Competition. As usual with these things, we're extending the deadline. The original deadline was March 31st. We're extending the deadline to April 18th. If you would like to submit, you can send an email to openvtprize@gmail.com. There's also a QR code here that will take you to it. If you have any trouble, let us know. Any paper with multicellular modeling in it that was published after January 1st, 2023, and where the code for the simulations has been made publicly available is eligible. You can use any framework or no framework. There also is the Biomodels Model of the Year competition. We've had the winners of that Biomodels competition from last year give talks in this series. They've also extended the deadline for their competition. The solicitations are not identical, but are similar. And so many people might be eligible to apply to both. Biomodels also does a lot of network modeling of various kinds. And so if you have a recent network model that you've distributed through biomodels or another public database, I'd encourage you to apply to that as well. It doesn't have to be a network model. They'll take any model that's publicly distributed. At the annual Society for Mathematical Biology meeting this year, we are going to have a GLIMPRINT mini symposium on immune digital twins. It's going to be 3-session mini symposium. Potentially, there may be room for 1 or 2 additional presentations. So if you would are planning on attending SMB and you would like to present in the general broadly conceived area of modeling related to the immune system and digital twins, please let Tom Helikar know as soon as possible, since he's the lead coordinator for that effort. There's also going to be a multicellular modeling workshop on reproducibility at SMB that you can register for at any time. If you're interested in that, you can come to me with questions. So I don't know how many people in the room here are going to be at the SMB meeting, but if you are, this one is the day before the main meeting starts. The GLIMPRINT ones will be during the main meeting, although we don't know the dates exactly yet. There's also coming up the 4th International Summer Institute on Network Physiology. The application deadline's officially passed. I don't know whether we can still apply for that. Moffitt is hiring faculty in the general area of multiscale modeling related to cancer. That's a good opportunity. Let people know about that. We don't have anybody booked for April 17th at the moment. May 1st, we have Marcus Covert. May 15th, we also have a slot. You've heard me say it many times. Feel free to suggest yourself, your graduate students, your postdocs, or anybody who has spoken or written that you think is interesting. Please let us know who you would like to hear from. And Lorenzo in particular is very good at getting people on the schedule, so please do help us fill the schedule going forward. Especially if anybody has a short time interest in speaking on April 17th, please do let us know. We'll have a steering committee meeting tomorrow at 11 a.m. If you think you should be invited and you aren't, email me or Tom and we'll get you on the list. Are there any other announcements before we move on to the seminar? Okay, today we have a single talk by Ramon Ortiz Catalan on multiscale Monte Carlo modeling for radiation therapy applications. And I will turn the screen share over to you.
Okay, so yeah, I'm Ramon Ortiz, postdoctoral researcher at the Department of Radiation Oncology of the University of California, San Francisco. And yeah, I'm going to talk about part of our kind of ongoing work on multiscale Monte Carlo modeling for radiotherapy applications. So I would like to start by very briefly introducing the fundamentals of radiotherapy. So radiotherapy is the medical use of ionizing radiation for treating cancer, mainly malignant solid tumors. But radiation can be also used for other forms of cancer, like some type of leukemia or for treating benign conditions. So the delivery of radiation treatments can be performed with external radiation sources, which is the most common form of radiotherapy, but also using radioactive implants within the tumor, or even using radioisotopes attached to specific agents that target the specific cell receptors. So radiotherapy can be used as a standalone primary treatment to eradicate the tumor, as a neoadjuvant therapy, for instance, to shrink the tumor before surgery, as an adjuvant treatment after surgery to kill tumor cells that may remain in the tumor bed after the surgery, and also in combination with other cancer therapies such as chemotherapy or more recently, immunotherapy. So the biological basis of radiotherapy is that ionizing radiation, when it interacts with matter, undergoes different physical processes like ionizations or excitations. And these interactions can directly damage the DNA if the interactions occur within the DNA components, like the DNA strands, and also create indirect DNA damage when the radiation interacts first with water molecules and then create reactive free radicals like the hydroxyl radical, which can diffuse and eventually interact with the DNA, causing, for example, oxidation of the bases, break of the backbone, or cause the strand breaks. So these two mechanisms of action can lead to different forms of irreparable damage. The most common is clusters of DNA double strand breaks, eventually leading to the cell death. So the fundamental principle of radiotherapy is to maximize the damage to the tumor cells while minimizing the damage to the normal tissues. And there are different strategies to achieve that. For example, using a specific particle types or different energies or different radiation field arrangement to maximize the dose at the tumor while minimizing the dose that is received by renal risk. And another common strategy is the fractionation of the treatment. So instead of delivering a single fraction of radiation, the treatment is divided in different scales of fractions to exploit the different radiobiological responses of different cell types, which can be summarized with the R's or the so-called R's of radiobiology, which are the repair, reoxygenation, redistribution, repopulation, and intrinsic radiosensitivity. So with this fractionation mainly allows that normal tissues can repair the sublethal damage between fractions more efficiently than tumor cells. The regions or hypoxic regions in the tumor, which are more radioresistant, can become reoxygenated between fractions, making them more sensitive to the subsequent irradiation. Also, tumor cells can move to more radiosensitive phases of the cell cycle, like G2 or M phases between these fractions. Also, between fractions, damaged normal tissue can repopulate. And with the fractionation, we can also take into account the intrinsic or inherent radiosensitivity of different tissues that may be early or late responding to radiation. And recently, other determinant factors has been added to these principles, such as the reactivation of the immune system after the irradiation. And some techniques are based on exploiting this effect by delivering fractions separated by several weeks to allow that the radiation-induced immune response contributes to the tumor control besides the action of radiation. Okay, so in this context, our main research focus in our group is the multiscale modeling of radiation effects with Monte Carlo. And in particular, the development of Monte Carlo simulation tools to model the transport of ionizing radiation through the patient in the context of radiotherapy, and also the application of these tools to develop and optimize radiotherapy techniques. So we are developers of the TOPAS and TOPAS-nBio codes in collaboration with our collaborators at Harvard Medical School and MGH, with whom we have been developing this code for over 15 years, thanks to the NIH funding. So in short, TOPAS is a Monte Carlo toolkit that simulates the detailed transport of ionizing radiation into a patient. So basically, this toolkit provides a reliable, experimentally validated Monte Carlo tool for medical physicists, radiobiologists, and clinicians in the context of radiotherapy. And importantly, as we will see later, it includes a specific user-friendly system of parameter control files to build the simulation files that allows the user to set up the simulations in plain English without the need of coding in C++, for example. So just to summarize the uses of TOPAS, one of the main uses in radiotherapy research is the calculation of dosimetric quantities at the patient level, quantities that can be typically used in radiotherapy treatment planning and that associate with treatment outcomes. And for that, TOPAS also incorporate biological models for predicting or assessing the treatment outcomes based on the distribution of these physical dosimetric quantities. But when modeling the interaction of radiation with matter at patient scales with millimeter-sized voxels are used, a common approach is to improve the efficiency basically is to group multiple reactions together and processing them as a single event. And this is called condensed history Monte Carlo for radiation transport. But when it comes the study of biological processes that may happen at cellular or subcellular level, which involves the micro and nanoscale, for example, at DNA level, we need to consider the individual interactions of radiation with matter, as we can see here in red, in terms of processing them as a single event. And this approach is called track structure Monte Carlo. And this is important, for instance, when studying how radiation types or how different radiation type of different energies can lead to different biological outcomes. So for example, here we have the spatial distribution of ionizations or interactions in yellow produced by typical X-rays using radiotherapy. So we can see that this distribution, the spatial distribution is nearly uniform compared to the one for heavy charged particles like carbon ions, which are also used in radiotherapy, which cause a higher density of ionization along the particle track. So when these patterns overlap with the DNA strand, for example, the degree of complexity of the damage produced is different, and due to this clustering of interactions. And this different complexity of damage can lead obviously to different repair efficiencies and eventually different levels of cell death and different treatment outcomes. So for the modeling of the biological effects of ionizing radiation at micro and nanoscale, TOPAS includes an extension called TOPAS-nBio. And the main use of this simulation tool or the simulation extension is the simulation of the interaction of radiation with a geometrical representation of cellular or mainly subcellular components. And this includes the simulation of the physical stage and the main physical interaction on nanometer scale, as I showed in the previous slide, but also the physiochemical and chemical stages, including how the radiation creates radical species and how they diffuse and how they recombine between each other to form new chemical species. And all of this also includes the analysis of the spatial distribution of these physical and chemical interactions and how they interact with relevant biological geometries, for example, DNA, to compute the yields of direct DNA damage and indirect DNA damage. So apart from that, TOPAS-nBio also includes biological models such as DNA repair processes or processes of misrepair of this damage, which can also account for the chemical changes in the microenvironment. And finally, this tool also includes biological models for the prediction of the different biological endpoints, such as the risk of secondary cancer or the probability of tumor control. Well, this is just an example of the validation again, experimental data, for example, of the quantities that can be calculated in TOPAS-nBio, such as like nanosymmetric quantities, the yield of single and double strand breaks, and also in different conditions, such as different oxygenations and also different temperatures. So basically, with TOPAS-nBio, we are able to model the radiation transport and biological processes at subcellular level, mainly at the DNA level, which is the main biological target in conventional radiotherapy. And also we can find association between this biological process or these biological outcomes as function of dosimetric quantities such as different particle types, different particle energies, different dose rates or other. For example, we can associate the yields of, I don't know, complex irreparable damage following the irradiation with a specific particle type. And then using these associations in TOPAS, we can model the treatment outcomes on patient level by using these associations and the distribution of these dosimetric quantities in a real treatment at the patient level. But while in conventional radiobiology, the main target is the DNA, when it comes to the development of new emerging techniques such as spatially fractionated radiotherapy or flash radiotherapy, what we are aiming is to exploit the non-targeted effects of radiation, such as the immune response, bystander effects, or other cell signaling effects that happen at cellular and tissue scale. And due to the level of detail of the simulations with TOPAS-nBio, the feasible scales we can explore are nano micrometer scale and from picoseconds to up to a few minutes. So for exploring the non-targeted effects of radiation in the context of developing new radiotherapy techniques, it is required to add another scale in terms of space-time, but also in terms of the type of mechanisms that we can simulate with these softwares. And here is where agent-based software, and in particular Computational 3D, fits into our research, and where we can benefit from the capabilities, potential uses of Computational in this context, such as simulating biological mechanisms in individual cells and simulate or model how they interact with each other and with the environment. Well, maybe this is not the most appropriate mechanism in this context. I think it was a nice animation from the Computational group to illustrate this change of scale I was talking about. So in this context, one of our research focus now is to link these softwares to be able to add these new scales to our modeling efforts for developing radiotherapy techniques. And in this context, we have developed a preliminary version of a framework that allows this multiscale simulation. This framework was called preliminary TOPAS tissue. And what it does is to couple TOPAS and TOPAS-nBio with Computational. So in short, that allow us to compute in TOPAS a given relevant physical, chemical, or biological quantity, such as the yield of double-strand breaks in individual cells, for example, and then continue the simulation of subsequent biological processes at a larger scale in Computational. So just as a proof of principle example of this, of the use of this framework, we model the clonogenic assay, so basically the in vitro cell survival of a PC3 cell line after X-ray irradiation. And for that, what we did was to compute the dose per nucleus in a kind of realistic cell population geometry, an in vitro cell colony, and in TOPAS, and also the resulting double-strand breaks per cell nucleus. And then with this information in Computational, what we did was to simulate the cell repair or the DNA damage repair and the cell response to this DNA damage in each individual cell in Computational. So in particular for the repair of DNA damage, we implemented the two-lesion kinetic model, which is widely used in conventional radiobiology, and we couple it with a published model of cell response that is based on the oscillation of the p53 protein as function of the double-strand breaks that are being repaired. So in essence, with this study, with this proof of concept example, we were able to reproduce the response of a multicellular system from, let's say, first principles. So we simulated the step-by-step interactions between the physical, chemical, and different biological processes. And this is important because we were able to consider the individual blocks of this response of living tissues, or in this case was in vitro, of cells to radiation. And this allows us in our research to consider different conditions at these stages. For example, in the physical stage, we can consider different particle types, different energies, different ways of delivering this radiation. And also this was built in a modular way, so we can also add different repair models that characterize different cell types, or also add different models that simulate other biological mechanisms of interest in radiotherapy research. And we opted to develop this framework with TOPAS in particular, and TOPAS-nBio, because there are several unique capabilities that are built in the software, especially in TOPAS-nBio, in terms of the simulation of and the simulation, and also the scoring of relevant physical, chemical, and biological processes that we are interested in the context of our research. And also we wanted this framework to use the user-friendly TOPAS parameter file system to be more accessible to the users of TOPAS. So just to briefly describe this parameter file system. So each parameter file consists of simple text lines specifying each of the simulation parameters in the simulation. And the format is, at first we have basically the type of variable, which can be as double, integer, a vector of booleans, etc. And then this is followed by the part of the core they belong to, for example, geometry, scoring, material, graphics, time features, and many others. And then we specify the parameter name. There are like many built-in parameters in TOPAS already, and also the value of this parameter. And the main advantages of these systems within that are that there is no need for coding experience, since it uses English language. The parameters are order independent to avoid like user errors. It has a hierarchical structure, so it allows to split up the simulations in different files, one depending on another in a hierarchical way. Also, I think it's important that it has a inherent system of units. So for instance, we can define a parameter in millimeter or centimeters, and the code internally will convert the parameter to a reference unit to make the different parameters depending on each other more consistent. And also the code internally checks for the right parameter and units to avoid the user errors that may create like some ambiguous situations. For instance, TOPAS won't allow to define the dimension of a geometrical component in degrees or with a Boolean, but instead we'll throw an error-specific method and we'll stop the simulation. So, yeah. So we created this framework to TOPAS tissue using the same system. And what we did was to add specific parameters that are defined in CompuCell. So for instance, here we have an example of setting up of a simulation lattice, the conversion from Monte Carlo steps to time and voxel size to real volume. We can also define the different chemical fields that will be simulated, and also the use of different models for different cell behaviors. For instance, currently we have implemented one or two models for damage assignment, damage repair, and cell response, but we are working on adding more or implementing more, not only for these mechanisms, but also for other signaling effects, for example. We can also define the characteristics of each type of cell. For example, we have here an hypoxic cell. We can define the dimensions, the growth rate based on a specific growth model. Also, we can define if this cell is sensitive to a specific chemical field. And also we can define things like the transition of one cell to another based on the availability of the concentration of, of five specific fields, which are all capabilities or parameters that are already implemented in Computational. So what the framework does is that from these parameter files, the framework will create TOPAS and Computational simulation files in their inherent language, and will run TOPAS and Computational at the specific times specified by the user, transferring the simulation results and results from one software to another. So for example, going back to one of the first slides, talking about how we can minimize the damage to normal cells with the fractionation of the treatment, we can use TOPAS to compute a given dosimetric quantity in a given cell configuration, and also, for example, the double strand breaks. And from the result, we can proceed the simulation with Computational of other mechanisms, and at the time of the second fraction, the current status of the cell configuration and also the cell microenvironment will be transferred back to TOPAS, which will take into account these changes to simulate the subsequent irradiation. Because another reason we wanted to create this framework is that we were not only interested in doing one simulation with one software, saving the results, and then starting the simulation, an independent simulation with the other software, but we also wanted to simulate or model in a quasi-parallel manner different processes in the different softwares that may overlap in time. Because, for example, in some techniques, such as radiopharmaceutical therapy, the radiation is delivered during several days at very low doses, which overlaps with in time and influence many other biological processes that may happen since the start of the irradiation. So again, what TOPAS can do is to do this transfer of information and run the different codes at regular times in a quasi-continuously manner, if we want, based on the time scales of each of the processes that are being simulated and the characteristics, for example, of the radiation delivery. Well, this is just an example. It's basic example, but it illustrates very well what I am talking about. So here we have like tumor cells forming a tumor with a vessel that is supplying oxygen. And this tumor starts growing until we deliver the first fraction of irradiation. So at this point, which was a point specified by the user, the status of the simulation in Computational was transferred to TOPAS. Then we perform an irradiation, and then the information about the dosimetric quantities that each cell received and the double strand breaks that were produced is transferred back to Computational, which simulates the cell repair, in this case, the cell death due to this radiation damage. And at some point, the tumor will start repopulating again until we deliver the second fraction of radiation using the same methodology again, and so on. Sorry. So using this framework on basically TOPAS-nBio and Computational, we were able, for example, to study how the tumor growth and the vascular redistribution as the tumor grows contributes to the preferential effect of microbeam radiotherapy. So in short, these radiotherapy technique, which is like microbeam radiotherapy, they will delivers a very inhomogeneous dose pattern. Well, this is like the lateral distance of the dose profile with peak doses, with lethal peak doses of hundreds of rays, which is way higher than what is using conventional techniques, which delivers like typically 2 rays per fraction. And these peak regions are separated by low dose, valid doses of 5 to 10 grays. So in preclinical studies, these patterns appears to result in a higher tumor control, but a reduced normal tissue toxicity as compared to conventional techniques. And the radiobiological mechanisms behind this response or this beneficial response are not fully understood yet. But among others, a potential mechanism in this technique is hypothesized to be the preferential effect in tumor vasculature as compared to normal tissue vasculature. So in this specific study, we wanted to evaluate how this dose delivery pattern may interplay with the rearrangement of the vasculature at different stages of the tumor growth and how this can affect the perfusion of tissue, of oxygen to the tissue and the tumor control. So to generate the simulation geometry, we first took segmentation images of the vasculature of the brain of a mouse, and we focus on the part of the in a part of their brain in the right hemisphere, which is typically the implantation tumor site in preclinical studies. And then we imported this geometry in TOPAS and Computational. So to import this geometry, we created a tool that is publicly available now, because although TOPAS and Computational have the capability of importing these geometries in a voxelized basis to this software, the main problem we face is that we needed not only the geometry in a voxelized geometry, but also we wanted the clustering of voxels that conform the same cell, because in radiobiology, the main associations between dosimetric quantities and biological effects are made in a cell or cell nucleus basis. So for that, we develop a, let's say, user-friendly tool that was written in Python and C++, that based on the user input, it can crop, resize microscopy images or segmentation images. But most importantly, it can create clusters of adjacent pixels based on the user specifications that will represent an individual entity such a cell. So at the end of the day, the output of this tool is PIF and TH ImageQ file, which are compatible with Computational and TOPAS respectively. And in each of these files, each cluster of boxes that represent a cell has its own unique identifier and represent this entity. So then we could compute first in TOPAS a given dosimetric chemical and biological quantity in a cell basis, and then assign a given behavior in Computational based on these quantities computed in TOPAS to continue the simulation of subsequent mechanisms. Well, just to mention that we validated this tool by comparing with different microscopy images. We compare like basically the distribution of voxels and also the number of individual cells in the original and the generated geometries, and in general we found a good agreement. Yeah, well, this was just to mention this tool because maybe it is useful or interesting for someone here. So once we have this geometry defined in a cell basis in Computational, we simulated the rearrangement of the vasculature of the tumor rows. And for that, we place a tumor cell at the center of the volume, and we assign the cell the size and behavior of a glioma cell in terms of like the cell division. And the cell start multiplying, dividing, the tumor started growing and modifying the geometry of the vasculature. And for this, we adopted a model implemented by the Computational group in a previous publication. And basically, we consider the autocrine secretion of a chemotractant field and the vascular cell elongation as the main actors for the modeling of the vascular remodeling. So in particular to this model that includes a VEGF like field secreted by vascular cells toward the medium, we added constraints for cell elongation with data found in the literature. Sorry. So at the end of the day, we had the distribution of the capillary at different stages of the tumor growth and also in the normal tissue scenario. So the idea here was to see how the damage of a portion of the capillary due to the very high doses could disrupt the flow of oxygen through the vasculature and how this affected the perfusion of oxygen to the tissue. And for that, we needed the flow of oxygen through the capillary rather than assuming that each vascular cell had a given oxygen concentration that could diffuse into the cell media. So in Computational, we modeled the oxygenation with the partial pressure of oxygen, or PO2. Let's assume that this is like simplistic representation of a capillary just for demonstration purposes. So to simulate the flow of oxygen through the capillaries, we used a diffusion field that was restricted to vascular cells, meaning that this field could only diffuse along the vasculature, but the diffusion coefficient in the rest of the lattice was zero. And we call this the vascular PO2 field. So to simulate the direction of the blood or the oxygen flow, sorry, we introduced donor and acceptor cells at the two opposed boundaries of each capillary with the size of the simulation lattice. And donor cells had a constant value of 90 milligrams of mercury, and we assign a value of 0 to the acceptor cells to mimic kind of like the oxygen drainage. So the vascular field diffuses along the vascular cells between these boundaries, and we did that for all the capillaries in the simulation. With this implementation overall, we found a mean partial pressure of oxygen in the vasculature that was similar to values reported in the literature. It was around 2 to 30 millimeters of mercury. So the oxygen flow to the medium, in this case, was model coupling this vascular field to another field that we call cellular PO2 field, which can diffuse throughout the entire tissue. So what we did was at each Monte Carlo step, the cellular PO2 field in vascular cells was updated to match the vascular field value, and then it was allowed to diffuse freely. At the same time, the vascular field was reduced based on the amount of oxygen that diffused from the vascular cell to the rest of the lattice in the form of cellular PO2, which was estimated by the change in cellular field compared to the previous step. So basically, this approach separated the oxygen transport, let's say, in two stages for the diffusion through the vessel and then the diffusion to the surrounding tissue, while keeping these two fields dynamically linked and dated at each simulation step. So overall, for example, the value of the vascular field in each vascular voxel was the result of a change due to the diffusion along the capillary and the loss of the concentration of oxygen that was diffused into the medium in the cell in the form of a cellular field. And this approach allowed us to simulate the disruption of oxygen, or at least approximation to the disruption of oxygen flow when vascular fields were killed by the high peak doses during irradiation. So for example, for this proof of principle example, we can remove two cells that we assume that are killed by radiation from the capillary. And we see that cells that are connected only to donor cells maintain a stable value of this field, while cells only connected to the acceptor cells lost oxygen quite rapidly due to the drainage, due to the oxygen flow and the diffusion of oxygen toward the medium, and cells that had no connection with any sides of the cell lattice with the capillaries lost the oxygen more slowly, only due to the diffusion towards the medium. So maybe for the sake of time, I will skip this, but just to mention that we modeled the death of vascular cells based on the mean dose that was received, mean dose to the cell, and the probability of vascular cell death measured experimentally, and we measured the cell outcome based on the different levels of oxygenation in each of the cells, and with the output of outcome of normoxic dividing cell, hypoxic no dividing, or necrotic. And also we model the consumption of oxygen by cells based on values found in the literature. So we radiated the different geometries, that represent the different stages of the tumor growth, and also the normal tissue with one fraction of microbeam radiation. And we follow the progression of the oxygen levels in the vasculature and in the cell lattice. And summary, we found that an association between the decrease in the spatial uniformity of the vasculature as the tumor grows, and a decrease in the perfusion, and decrease in the perfused vessels, and also a decrease in the oxygenation of the tissue after microbeam irradiation. So, in summary, we report that that as the tumor grows, it displaces the vasculature towards the tumor contour, and the uniformity of the capillary coverage within the cell lattice decreases, which is something that was expected. But interestingly, the number of vascular cells that were killed by radiation itself was similar in all of the scenarios, but the number of unperfused vascular cells and the levels of cell oxygenation decrease as we go towards more advanced stages of the tumor proliferation. And even the relative change between the post and pre-radiation and oxygen levels was dependent on the stage of tumor proliferation, what we can see here. So in terms of cell outcome, we can observe a similar portion of cells, normal and tumor cells killed by radiation, which was around 20. But interestingly, the oxygen depletion after irradiation that I showed in the previous slide affected the fraction of hypoxic and necrotic cells, depending on the tumor stage. So in normal tissue and early stages of the tumor, cells remain well oxygenated, but at later stages, microbeam radiotherapy led to a higher fraction of hypoxic cells, up to 70%, and an increase also in the number of necrotic cells, which was up to 10%. So overall, these results align well with experimental results that show that there is a tumor-specific hypoxia and necrosis after microbeam irradiation, while the normal tissue remains with normal levels of oxygenation. But the results kind of provide a potential cause for that, which is that the redistribution of the vasculature as the tumor grows, decreasing the uniform distribution of this capillary, and the interplay of this rearrangement with the highly inhomogeneous dose delivery microbeam radiotherapy may lead to hypoxia in the tumor, which may slow down the proliferation, and also necrotic regions, which potentially can contribute to the tumor control, but while preserving the normal tissue. So just to finalize, I would like to mention some of the limitations of these works. We only focus on the the impact of the vascular redistribution, the spatial redistribution during tumor growth on microbeam radiotherapy outcomes, but we did not account in for the different radioresistance in the vessels in the tumor or normal tissue. So maybe future work can include that. Also, we can simulate or model more accurately the oxygen diffusion, the vascular damage, not repair, but also the disruption of this oxygen flow when vascular cells die. Also, it would be interesting to look at the spatial distribution of the oxygen levels and the hypoxic and necrotic cell regions, which was not evaluated in this study, and well, probably other potential improvement. So I leave here the article, the publication, in case anyone is interested in checking that. That was all. I would like to thank the Computational collaboration for the invitation. Here I leave my email in case that someone wants to discuss further the results, or there are things that we can do together in the future. So yeah, thank you very much.
Okay, thank you very much. Time for some questions. I certainly am interested in the model specification language that you've done, but I was going to focus maybe on the last bit where you do the oxygen transport. And in this case, you're using a diffusion model of oxygen transport as opposed to an active flow transport model. Do you think that that's significant here? Would it be worth putting it at explicit bucket brigade where you actually move the oxygen down the vascular tree? I suppose if it's interrupted, you don't have flow. But I wondered if you wanted to talk about that, maybe a few words.
Yeah, I think that, well, in this case, maybe I didn't mention, but we just consider, since these microbeams are micrometer, they have a width of micrometer, we just study like 0.7 millimeters cubic volume, 0.70707, for like computational efficiency. But I think it will be interesting to adopt other methods for oxygen transport when we take into account maybe larger regions of the brain or other tissues, because in that way I think that we will see differences in the oxygenation of the individual vascular capillaries depending on their arrangement and also their size. So yeah, I think that for studying larger or even this, but I think this will have an impact when we study larger irradiations, for example, whole brain irradiations.
Well, I'll ask another one. Since bystander effect is very important here, you're modeling bystander effect in the form of damage to the capillaries and therefore hypoxia. Would it be practical or interesting to try to include immune cells in this simulation at the multicellular level? And if so,
Yeah.
what cells would you pick to begin with, and what signals would you start with, maybe?
Yeah, the response of tissues to this specific pattern is hypothesized to be a combination of different effects, and one of them is bystander. Another one is the immune response that is triggered by these high doses at the specific regions of the tumor. So, but none of them are well modeled yet. They are just hypotheses. There are like some experimental hints that they can play a role. So I think that first it would be interesting to model them or characterize them in control conditions, and then maybe put a model together with all of them, because I think it will be quite complicated to, without any hint of how they are modeled in this kind of like very high dose regions, and also in this low value region, and what happens between the communication of cells that are here and here. I think it will be interesting of studying them individually first and then put them together. But yeah, I think it will be very interesting to attenuate kind of models like immune responses.
Michael, did you have a question about the model specification language? Maybe we could go back and look at it together, and you could maybe ask a question or two about that. So maybe you could just walk us through this. What's the first column in the model spec is what here, where it says
Yeah,
BDI? Why don't
so
we
this
walk
is
us through the spec?
first we have the type of variable. So,
Okay.
well, the type of variable, this will be an integer, this will be a
Okay.
unitless value, that will be a
Okay.
double. This is a double, so doubles have units by definition.
Mhm.
This is a vector of doubles. We have not here, but we have also booleans like here. And then since this was implemented in the already built TOPAS parameter file, we created a new part of the code that was called CC3D. Instead of like in the previous one, we have geometry, scoring, material graphics, and other. And then we have, in this case, it was like very preliminary, but we have different parameters that are built in these systems that then will convert these parameters into the into Computational simulation files for the simulation in Computational. Here we have something that maybe it's more complex, but first here we have again the type of variable, the part of the module, they will be analyzed, and then the type of cell. And then in this case, what we did was the second part, if it was a field that it's defining this list of fields to
Right.
consider, it means that this parameter, it's a parameter that will related to the oxygen field that will affect this type of cell in particular. And then based on this, we can also define another, or we'll just put this one as an example. So this transition, which is a process, will be dependent on this field and will affect this type of cell, which will lead to this other type of cell.
So that would then basically compile to a line of Python that would be in a loop that checks whether that condition is true for that cell type at a given time.
Yeah, yeah. Yeah, in this case, I think we modeled with the Python code in Computational.
Right. All right. So, because there's been a lot of discussion about whether it's practical to write a rule set and model specification that looks like this. I mean,
Yeah.
CompuCell
Well,
historically, and the fact that you've done it is quite impressive to me.
also I wanted to mention, I don't know if the impression that we were not pretending to replace the Computational tools. It was just like we wanted the users of TOPAS, which are familiar with parameter system file, to have more accessible, more like easy access to this, to modeling with Computational at the first time when using the combination of TOPAS and Computational. But this was just like to be more user friendly with our users of TOPAS, not to replace the Computational language.
Oh, well, but we're very interested in the possibility of having a language like this that you could use to specify, for example, CompuCell or Morpheus, which is another programming framework that uses the same approach, or Artisto or Chaste. So there are at least 4 frameworks that generate the same kinds of simulations. Each one of them has a different model specification language. And so having a high level human readable model specification language that you could then compile into the various specific languages is very attractive.
Yeah.
Yes.
Also, the idea of putting this like this is at some point we were also thinking that maybe there were other processes that we can simulate with other software. Like I don't know, we were thinking at some point with B-cell. So it was a way to not to have to write needs a specific software when using TOPAS and coupling TOPAS with this software. So it was more like for the users to have an easy access to this framework.
And could you maybe just for a second show how is the time sequencing done? So if you want to evaluate, the damage at each region. So CompuCell would then, is it pre-computed or does CompuCell then call the micro model and say, and to evaluate the amount of damage in a given voxel or in a given cell? How does that work?
Yeah, this is something that we haven't went further because we were discussing how we should do it because TOPAS is multi-thread. So we are still discussing how we need to do this transition. If we need to put a pause in the simulation of TOPAS and then transfer the results to CompuCell, and then do like a pause each simulation and transfer the information. We are checking if this will be possible, but it's still under discussion, or just like have like a software that is always running and just to transfer the information and it's a specific Monte Carlo step. For example, if CompuCell takes more time of the simulation in terms of like the time scale of the biological process, and we can assume that a given radiation is instantaneous as compared to this time scale, we were thinking, okay, so we just like post the CompuCell, we run TOPAS, we close TOPAS, and then we transfer the result. So it's something that was under discussion still because, it's more like the architecture of the softwares and how we can do it more efficiently. And I am not an expert on kind of things.
No, it's fine. It's quite interesting to me to think about how to do the scheduling and wrangling of the data back and forth. And to me, this is a very impressive effort that way. Okay, well, I want to thank you for presenting. I'm looking forward to hearing how this develops in the future. Maybe we can have you come back in a year and tell us how it's gone. Thanks, everyone. I'll see people at the next meeting, and I appreciate you coming today.